All right, so here in chapter 11, we're talking about the mechanisms of microbial genetics. And specifically in 11.1, we're just briefly going to go over the functions of genetic material. And this is a very short section of the chapter to begin with. And also, we have just gone over this material. So this is kind of a little bit of a refresher, just recalling that one, genetic material is passed from parent to offspring. So genetic material is passed from parent to offspring, which we all know, but where it gets a little bit different here is that when we think about the parent and the offspring, when we think about eukaryotic organisms, and we think about, let's say, humans specifically, it's very easy to know that, the DNA comes from the parent and is passed down to the child. Or otherwise known as the offspring. But then also keep in mind when we're talking about prokaryotic organisms, it's the same. It's from the parent to the offspring, even though it is just binary fission or budding or any of those different options, mostly binary fission. We're still talking about that genetic material is going to be passed from parent to offspring. And then two, that it directs and it regulates the construction of proteins. So it directs and regulates the construction of proteins, and really all functions. So then this is where we get to our central dogma of biology. So central dogma of biology, which is basically that DNA is going to encode for messenger RNA, which then in turn codes for a protein. So our DNA, and then we go through transcription to code for our mRNA. Then we go through translation to get to our protein. And so we'll be following along this path. This is what's called the central dogma. It goes from DNA to RNA to protein. So DNA through transcription to RNA through translation to protein. And then the second kind of piece of information that came from this 11.1 section is just the difference again between genotype and phenotype, which we just went over in 10.4. So genotype again is the genes, the full collection of genes that a cell contains, and remember that this remains constant. And remember that the phenotype is the observable characteristic. Observable characteristics. And that's based on the genotype. And then remember the example, a really easy example to remember because we've seen it in the laboratory, is that we have a genotype in Serratia marquescens. And Serratia marquescens, if we put it in one environmental condition, say at 30 degrees C when we incubate it, the other environmental condition is 37 degrees C. When we have it at 30 degrees C, then it ends up being pink when it is growing. All of the colonies are pink, versus at 37 degrees C is just the typical cream colored. So really that's all we kind of get from 11.1, going over some things that we've already really should be very, very clear to you at this point. When we get to 11.2, then we start to talk about DNA replication. And a lot of this may be clear to you as well, except we are going to talk very specifically about bacterial replication. So this piece here, that DNA replication is called semi-conservative, or is semi-conservative, should be not a new concept to you, but semi-conservative remaining that it's going to result in 2 DNA molecules at the end of replication, and then each one has a parent strand of DNA, and then also has a newly synthesized strand. So if we start with our DNA, this is our double helix here, then it's going to separate. When it separates, we have our initial parent strand, so this is the parent color here, and then it's going to separate, and then they're going to have one parent, and then we go through replication to make the second strand here. Actually, I could put that on the other side. There we go. The second strand here, and then when these go on to replicate, they're going to separate here and here and over here and here, and then they go through and then they make their copies of DNA. So every generation has a parent strand. So this was kind of the blue color here. So there's a parent strand here, and then there's a new strand. And then when we go down even further, then we have a parent strand here, and then the new strand. And that is semi-conservative. The second kind of refresher piece of information is something we spoke about in 10.4 as well, and that is directionality. So directionality is just reminding us that the nucleotides in DNA are incorporated into a growing strand on the 5' to 3'. So are incorporated into a growing strand, 5' to 3'. 5' to 3'. There we go. And this is important because we're going to have a leading strand and a lagging strand. And so when we look at these things, it's important to remember that we have to add it to a free 3' end. So no matter what we're doing, it's always going to be adding to that free 3' end. So we have to cleave the two end phosphate groups. Energy is put into that sugar phosphate bond, and then we attach it to that free 3' end. So cleaving the two end phosphate groups, meaning from ATP. So we utilize ATP, remember kind of what ATP looks like up here. This is our adenine. This is our sugar of ATP. There's an OH down here. And then we have our phosphate groups. And remember, so it's adenosine triphosphate, tri meaning 1, 2, 3 phosphate groups. When we use the energy from ATP, what we do is we cleave off the end 2 phosphate groups here. So we break that bond there. And the release of energy is going to be put into the building up of another molecule. In this case, we're talking about replication. So we're talking about building DNA. So that energy is then going to be put into the sugar phosphate backbone. So remember, we have a sugar, and we have a phosphate, and a sugar, and a phosphate, and then we have our nucleotides in the middle there. So when we put energy from this ATP breaking, we put that energy into adding on to this 3' end. And that's how we add our next nucleotide. Okay. So now we are going to take a close look at bacterial replication, or DNA replication in bacteria. So DNA replication in bacteria. And we are going to start by taking a look at what's called initiation. So as its name implies, initiation is the start of bacterial replication. So bacterial DNA replication. So when we replicate the DNA in bacteria, we start at what's called the ORI-C, which really stands for origin of replication, or ORI-C. So it's ORI-C, or the origin of replication. And then this is always a very specific nucleotide sequence. And what happens is, to initiate this DNA replication, our proteins are going to bind to this origin to begin replication. So the cell is metabolizing, it's happy, it's got lots of energy, it's going to go through replication, decides it's going to go through binary fission. So it's going to make a copy of its DNA. So then these proteins that we'll talk about in a moment are going to bind to the ORI-C, the origin of replication, and this is what's going to start replication. So some of the enzymes that we're talking about, or some of the proteins that we're talking about, Recall that our DNA is very highly wrapped up. Remember, we have these histone proteins or histone-like proteins, depending on the prokaryotic organism that we're talking about. If we're talking about archaea, for example, we are talking about bacteria here. So we have these histones, and the DNA is going to be very tightly coiled, and it's going to be coiled around the histones, and then it's going to be coiled between the histones, and the histones are going to be coiled around the histones. So it's all very tightly coiled. So the bacteria needs an enzyme to go in and uncoil this DNA. The way I think of this is one of these old school telephones. If you remember the telephone that has that kind of curled cord. And that curled cord, if you pulled on it too much over and over and over, it would start to get these really tightly wound sections. So this would happen with our DNA if we didn't do something about it. Typically in those phone cords, what you would do is you have to hold the cord and then let it unwind to try to separate it out, or kind of yank on it to pull it apart and relax those coils again. But this would be damaging to DNA. So our bacteria is not going to do this in the cell, so it's going to try to prevent this really highly twisted up and coiled DNA. It does this by utilizing an enzyme called topoisomerase 2, also called DNA gyrase. That is not how you spell that. Gyrase. So topoisomerase 2, or DNA gyrase. And what this does is it relaxes the strand. And the way it does that, so if we think about that telephone cord, and let's say it's all wound up like this, and in this case we're talking about it as DNA, this topoisomerase 2 is going to go down. So let's say the ORI C is right here, and so it wants to attach here and start replication. So what topoisomerase 2 does is it goes further on down the line, and it's going to nick the DNA. It's going to cut it. And then what it's going to do is it's going to untwist this or unravel this section and then stick it back together. So it creates a temporary nick ahead of replication. So replication is going to be going this direction, and in going this direction, it will also be going this direction. So topoisomerase will be making a nick over here and a nick over here, and it's going to loosen that, basically going in, and it's going to unwind this, kind of untwist it a bit so that it's not all wound up, and then glue it back together. And then when we go through replication, we don't have this intense twisting, and it's not going to then break the DNA. So the topoisomerase can be very specific about it. Go in, cut it, untwist it, and glue it back together. So topoisomerase creates temporary nicks ahead of replication. So the next enzyme we're going to talk about, or protein that we're going to talk about, is called helicase. And helicase is going to separate the DNA strands at that replication fork. So it's going to separate them. So separates DNA strands. And what it's separating are those hydrogen bonds, right? Because the hydrogen bonds, remember, we have our strands of DNA, and then we have our A's and T's and C's and G's in the middle there, and it's going to go in, it's going to separate those A's and T's and the C's and the G's, and then it's going to pull those two strands apart. So it's going to cut those hydrogen bonds. I always think of helicase as like a zipper. So we have the zipper that, and everything is kind of intertwined, they're attached to each other, and then we have the actual zipper, the metal piece, that goes down the middle and is going to unzip or separate those DNA strands. Now, those DNA strands are going to want to just go right back. They're going to want to create those hydrogen bonds. They're near each other. They're going to want to go right back to having that double strand and those hydrogen bonds being in place. So what we need to have then are enzymes or proteins that are going to hold those apart. And those are called single-stranded binding proteins. SSBPs. So the single-stranded binding proteins are then going to ensure that those DNA strands don't hydrogen bond back together. So they are going to stabilize the replication fork. So I'm going to go ahead and pull in a picture here so that we can take a better look at this situation. All right, so here is our image of DNA replication in bacteria, and I've kind of gotten rid of some of the labels here. This is an image from the text. So what we're looking at in the upper right-hand corner here is the zoomed out view first here. So our origin of replication is up here. So our ORI-C, remember our ORI-C. And you can see that we're going to have the direction of replication going in both directions, right? And this is what we call the replication bubble. Replication bubble. And of course, the reason it's called a bubble is because it looks like a bubble. So if you look at it underneath the microscope, a very high-powered microscope, you can actually see it looks like little tiny bubbles as it's going through DNA replication. So then we have this ORI-C, we have our replication bubble, and then we have our replication going in opposite directions here. So remember that we have our topoisomerase 2 or DNA gyrase that's going to relax the strand. So down here is where we have our topoisomerase 2 going to go in, and it's going to nick the strand, kind of unwind it the other way, and then glue it back together. And same thing up here. So it's going to glue it back together, but now it's relaxed those strands so that as we have this bubble and we're pushing and kind of separating these strands, just like that kind of telephone cord, if you took your finger and you started to pull it down that cord, straightening out those curls down at the end or twists down at the end are going to get tighter and tighter and tighter until they start to wrap around each other and they can eventually break. And again, that's why we don't want to do this with DNA. So our topoisomerase 2 goes down the way, and it's going to cut it, relax those tightly wound up curls, and then glue them back together. So then the second enzyme that we talked about, or protein that we talked about, is helicase. So on here, this right here, this purple triangle is helicase. And again, it's easy to remember because in the images it looks like a triangle, which reminds me of a zipper as well. So if we look at this smaller image in the upper right corner, again, so it's kind of like helicase comes in here and it's going to go this direction. And it's unzipping. It's breaking those hydrogen bonds between the two strands, and we have it over here as well, and it's going this direction. It's going to be breaking those hydrogen bonds. So we have helicase down here breaking those hydrogen bonds, and then right here, these guys are single-stranded binding proteins, and we have them on this side as well, single-stranded binding proteins. So these are the ones, I like the way that this image is because the helicase looks like a zipper, although I would have twisted it around the other way, and then the single-stranded binding proteins look like fingers, look like hands kind of wrapped around. There should be like a fingernail here or something. These are little fingers, little hands that are wrapped around, the arm would be this direction, and they're holding the strands apart. So these single-stranded binding proteins are holding them apart so that they don't go back together and create those hydrogen bonds again between the A's and T's and C's and G's. So these are our replication forks then. We have our helicase and we have our single-stranded binding protein, and then this is a replication fork, right? Because it looks like a fork, kind of, a fork that's missing a tine in the middle. But there's one here on this side, and then of course there's one over here on this side as well. You can see it up here in this image in the upper right-hand corner. This, of course, is a larger blown-up image at the bottom here, just showing us one replication fork within the replication bubble. Good. So now that we have, relaxed our strand, we've opened it up by utilizing helicase, who's unzipping the strands, and then those single-stranded binding proteins holding it apart. Now we want to get in there and start doing replication. And we do that utilizing DNA polymerase 3. And that is this one here. So DNA polymerase 3. So DNA polymerase 3, it also has what's called a sliding clamp, which is this here. So DNA polymerase 3 is going to bind the 5' end of a free nucleotide, a brand new nucleotide, to the 3' end on the growing strand here. So our DNA polymerase is this enzyme here, and it's going to be moving along, and it's going to be taking the free 5' end, or the 5' end of a free nucleotide, and it's going to attach it to a 3' end on the growing strand. So I just did the 3' end here, but it's actually up here because this is where our free end is. So let me write that down below here. So our DNA polymerase 3. So it binds the 5' end of the free nucleotide to the 3' end on the growing strand. So what this means is that we have to have a free 3' end on this growing strand in order to attach this new nucleotide. So if we think about the situation here, we have our DNA strand and we go in and we break it down the middle. Now what we have is our DNA strand here, but we need some way to start building our secondary strand. And our DNA polymerase III can only add a new nucleotide on a free 3' end. And at this point, we don't have a free 3' end, so we have to get a free 3' end. So we have a different enzyme that's going to do that, and that is called RNA primase. So RNA primase is going to make the primer, and the primer is important. The primer is an RNA primer. It's made by DNA primase, and that's because RNA polymerases do not need a free 3' end. So we have our DNA strand here that we've just opened up. And then in order for our DNA polymerase III to bind to that free 3' end, we have to have a free 3' end. So our RNA primase is going to come in and it's going to stick an RNA primer on the DNA, meaning that it's going to attach nucleotides here with our sugar and phosphate backbones here. And our nucleotides, these are, say, our nitrogenous bases there that are attached to the nitrogenous bases on the DNA here. I'm simplifying. So then this would be our primer, and it's made up of RNA. So I'm going to get rid of this primer I drew here. So now that we have the primer, now our enzyme, our DNA polymerase III plus the slider, or the sliding clamp, can now add to this free 3' end. So now this 3' end is available. Now DNA polymerase III can do its job by grabbing onto a free nucleotide here and attaching this 5' to the 3'. And this is what we see in the image up here. We don't see it up on top of this section. This is the leading strand. Now the leading strand needed a primer as well, but they just don't show it. So up here, there would have been a primer all the way in the upper right-hand corner is where I'm drawing. So this would have been the primer here, and this would have allowed the 3' end to be here, and then our DNA polymerase III would have attached and then could have added the nucleotides to these free 3' ends as it's going. And it continues to go this way. Now we do see the primers down here at the bottom. And so this green one here, this is RNA primase. And this is the enzyme that's now making, you can see this line here, I'm going to kind of curve it. This is the primer. So this arrow here, they're pretending are little RNA nucleotides that the RNA primase is adding opposite the DNA here, so that we have something for the DNA polymerase III to attach to and then add to a free 3' end. And you can see that here. What I like about this lagging strand down here at the bottom is it shows you kind of piecemeal how it happens. So this is the first stage here on the left-hand side. Let me get rid of some of these drawings. Okay, I can't. So here, what we have happening is our RNA primase is starting to make that RNA primer. And you can see that this is a 5' end here, which means that this is the 3' end here. And then you can see here, now we already have the primer, which is the green arrow or ribbon here, and then our DNA polymerase III with its sliding clamp here has attached to that primer on the 3' end, and now it's adding nucleotides. And the nucleotides, the DNA nucleotides, are in red, and then the RNA nucleotides here are in green. So now it's adding these DNA nucleotides to this free 3' end. Now, the leading strand and the lagging strand are an important concept because remember that DNA is anti-parallel. So anti-parallel meaning that one strand is oriented 5' to 3' and the other one is oriented 3' to 5'. So we have our leading strand, and our leading strand has continuous synthesization. It's continuously synthesized. So what that means is that we attach the DNA polymerase 3 to that original primer that we said all the way back up here in the right-hand corner, and then it's just going to keep going. And it can just keep going. Helicase is going to continue unzipping in this direction to the left in this image, and DNA polymerase 3 is just going to continue going down right behind helicase, adding on to the 3' strand or 3' end as it goes. So that's why it's the leading strand, and that's why it has continuous synthesis. It just can keep going, keep going with helicase. Now, the other strand is called the lagging strand, and it has discontinuous synthesis, and that's because it has to keep going back to helicase where it's opening up that replication fork and starting over. And that's because it has to add to that 3' end. So here we can kind of see at the bottom that if we look at this RNA primase here, and it's making the RNA primer, and we have the 3' end, then we can now, in this section here, we can see the DNA polymerase 3 with its sliding clamp attaches to that and adds the 3', or adds to the 3' end, all the way until it hits this primer here. Now this primer here was placed earlier, if we kind of go back in time this direction, but this was placed earlier because this at one point was the replication fork. So RNA primase came in right at the beginning, right where helicase was unzipping, and it added an RNA primer, 5' to 3'. Then our DNA polymerase came along, attached itself, and started adding these nucleotides, DNA nucleotides, all the way up until it hit the other end. Now, then helicase is going to continue going toward the left in this image. So then what happens to anything that's to the left of this primer? Well, it's open. So then what needs to happen is as we get closer to that replication fork again, back to the helicase, we have RNA primase is going to make an additional primer here. Then our DNA polymerase 3 is going to start adding to that 3' end all the way up until it hits the next primer, just like we see happening here where it hits this next primer. And so that's why it's discontinuous, is because it has to keep going closer to that replication fork. It has to keep going back, and it has to keep going back. Every time that replication fork keeps opening up further to the left in this image, we have to keep making primers to the left that are going to then add to the right, and then primers to the left that are going to add to the right, and primers to the left that are going to add to the right, until it keeps bumping into the previous primers that are added. So the leading strand only needs one primer, just right at the beginning. And then the lagging strand needs many primers. Now, once we have had sections that have these primers, and the DNA polymerase 3 is going to add to the 3' end up to the primers, then we get into the situation to where we can't have this RNA in our DNA, right? Because remember, it was an RNA primer. It's not made out of DNA. And since it's an RNA primer, we have to get rid of the RNA, and we need to get our DNA in there. So what we end up with when we have this primer and then this synthesis of the DNA and then another primer and synthesis of the DNA, we have created what are called Okazaki fragments. So Okazaki fragments are these small DNA sequence fragments that each have a primer. So they each start with a primer. So let me write that down here for you. So Okazaki fragments. So our Okazaki fragments are small DNA sequence fragments, each starting with a primer. And so that's what we have on this lagging strand. The lagging strand has many Okazaki fragments named after the person that found them. And then what we need to do is we need to go in, we need to take out the RNA, and we need to add in DNA and connect these fragments together. So how we do that is first we utilize DNA polymerase 1. DNA polymerase 1 is an exonuclease. What that means is that it's going to remove, exo is to remove or exit, nucleotides. Remember, ACE is an enzyme. So it's an enzyme that removes nucleotides. So what we're doing here is we are going to remove the primers, and it fills in the gaps. So with these Okazaki fragments, it's going to go in there, it's going to remove those RNA nucleotides, nucleotide by nucleotide, and it's going to replace them with DNA nucleotides. So if we look back up at our image here, we can see that again. This right here is our DNA polymerase 1. So DNA polymerase 1, you can see it's trying to show you here that the primer would have been this long, and then this is an Okazaki fragment. Now what this DNA polymerase is doing right here is it has replaced these nucleotides, the green nucleotides that were right here, for example, and replaced them with DNA nucleotides, which are red. So it's exonuclease, meaning it's taking out the RNA and it's putting in DNA nucleotides. So we have DNA polymerase going back in, it's replacing RNA with DNA, but then what we are left with is one last nick. It cannot create a bond between the last nucleotide that it replaces, so this nucleotide here, because DNA polymerase 1 can only add on to a free 3' end. So once it removes this RNA piece and brings in the DNA nucleotide, it is going to be able to attach that to the free 3' end of this nucleotide here, but it cannot attach it to the 5' end of this nucleotide of the next Okazaki fragment. It can't do it because it can only take the free 3' end and attach it, and then it leaves that free 3' end there. So then we have an additional enzyme called DNA ligase come in. So DNA ligase comes in, and what it's going to do is it's going to catalyze the formation of the phosphodiester linkage in that remaining link, meaning it's just going to attach those backbones. It's going to make sure that this right here gets taken care of. Where we have that last 3' to 5', it's going to glue those two things together so that everything is continuous. So all these Okazaki fragments are all glued together. So we can write that down here with our other enzymes. We have DNA ligase. So it catalyzes the formation of that last bond there, that last blank space. So as we've been talking about these things, we spoke about initiation. I mentioned initiation at the beginning, and recall that initiation of DNA replication was where we have this origin of replication, and we have our topoisomerase 2 is going to go down and relax the strand. Our helicase is going to go in and start unzipping the strands, and then we have this replication fork. We also have our single-stranded binding proteins are going to stabilize the replication fork by holding them apart, making sure they don't go back together. That allows our DNA polymerase to come in, DNA polymerase 3 specifically, with the sliding clamp to allow it to slide along the DNA, and it's going to attach to this strand, attach to the primer that has been made by the RNA primase, and then it's going to start adding nucleotides to that free 3' end. So all of that is part of initiation. Then we have elongation, and so elongation is actually the process. Elongation is actually the process of adding these nucleotides to the free 3' ends. So that's what DNA polymerase 3 is going to do once it attaches, right? So part of the attachment is the initiation, so it's initiating or starting it, and then elongation is just the continuous adding of these DNA nucleotides. Part of elongation is going through with these Okazaki fragments, so obviously making the Okazaki fragments is part of elongation because that's the adding of the nucleotides as they bump up to the primer. But then also the activity of the DNA polymerase 1, that exonuclease, removing those RNA nucleotides and then filling them in with DNA nucleotides, as well as the DNA ligase going in and kind of gluing together that last little cut. All of that is part of elongation. Now what we have at the end is termination. And there's really not a whole lot known about termination. What we do know is that we have concatenation. So the DNA strands are concatenated. And what that means is that, remember that our bacterial DNA is circular. So it's kind of all over the place, it's really long, but it is circular. It's not linear like our DNA. And since they are circular, then when they go through DNA replication, they're going through and we have this replication bubble and things are kind of starting and stopping and starting and stopping. What happens is they're interlocked. So that is what's called concatenation. So the circular DNA chromosomes are interlocked. So then what we have to do is we have to use an enzyme called topoisomerase 4 to break the DNA, pull them apart so that they're no longer wound around each other, and then glue them back together. So we have topoisomerase 4. And then that is the end of replication, DNA replication in bacteria. We had our initiation, elongation, and termination. So you should be familiar with all of those different enzymes and all the different parts of those, what they do and how they all piece together. You should also be able to draw, and in the text, this is all completed, this image here with all of, everything is labeled, just like I labeled it, but it's already in there typed. So make sure that you could write this out so you could label this type of image so that you know all of these different pieces. So we are going to take a look at both prokaryotic and eukaryotic organisms in this chapter. So we are going to talk about DNA replication in eukaryotes very quickly because you should be pretty familiar with this. So DNA replication in eukaryotes. So eukaryotic chromosomes are typically linear. So again, they're really long strands. Of course, they're twisted and turned and wrapped around things, but they are linear. They have an end, a start and a stop end. And each of them contain multiple origins of replication. So they don't have just one ORI-C, but they have multiple origins of replication. So what we end up seeing is multiple replication bubbles. So something like this. This wouldn't really end like this. There we go. So these are all replication bubbles. So replication is going to be happening inside here. So again, it's just like the forks that we saw. We have a fork going this way, going that way, and we have multiple replication bubbles that are happening. Now, some of the differences between DNA replication in bacteria and in eukaryotes is that we have DNA polymerase, but we don't have DNA polymerase 1, 2, 3, 4. Instead of DNA polymerase 3, we just have DNA polymerase. So instead of DNA polymerase 3. So in eukaryotes, we just have DNA polymerase that is going to be doing the job of DNA polymerase 3. Then we also have ribonuclease H, and ribonuclease H, also called RNase H. What it does is it does the job of DNA polymerase 1, and so meaning that it removes the primer. So it removes we'll say RNA primer, removes the RNA primer and then replaces with DNA nucleotides. So this is instead of DNA polymerase 1. So remember, DNA polymerase 1 in bacteria was what was going to go in, remove that RNA primer, replace it with DNA. Instead, we have what's called ribonuclease H, or RNase H. So in our DNA replication in eukaryotes, our replication fork is going to reach the end of our DNA, because remember, it's not a circle. So it's going to reach the end at some point down here. and then we don't have a primer to finish replication. So if we take a close look at what that means, if we have our 2 strands of DNA, remember when we go in, we have to start with an RNA primer, right? So these are, let's pretend, nucleotides. We've separated the DNA strand. These are separate DNA strands. We have our RNA primer here with our nucleotides, and then we're starting to add our nucleotides. So let's say this is our 3' end, this is our 5' end, and we're adding nucleotides here. This is continuous, the 3' end, so we're going this way. On this end, however, we have our primer. This is the 3' end on this primer, and we're adding our nucleotides this way. Now, when our ribonuclease H goes in, and it's going to remove these here, and then add in our DNA nucleotides here. So there would be an additional 3' nucleotide over here. We're going to be adding to the 3' along here. Over on this side, on this strand, if we go in and we get rid of that primer here, this is the end of our DNA strand. Remember that our enzyme, our RNase H, has to go in, remove the RNA nucleotides, and put in DNA nucleotides. Now remember, it has to attach it to a free 3' end. So if we take a look at this, it has no 3' end to add to, right? Because this is the end. So there's no free 3' end here to attach the nucleotide to, which is a serious problem, right? Because if we think about this, then if we just go along and now we have this new DNA strand that's going to go into the new cell, it's missing this entire chunk because that's where the primer was, at the very, very end of the DNA. So let's say, okay, that's fine. So it goes into the new cell. Now we're missing this chunk. So then these strands here are going to separate. So this one will go this way, this one will go this way. Now this strand here is going to be missing this entire chunk here. When it goes through its replication process, then it's going to have the exact same thing happen to it. So then it's going to be missing another chunk here because it doesn't have a primer for that same chunk here. So now it's going to be this much shorter. And then this happens over and over and over and over again. So we lose our nucleotides at the end when we have our linear DNA. And so this really does happen, and our DNA does have a way to try to protect our body from having this happen, or not protect it from having it happen, it happens, but protect it from damaging us as a human being and our functions. The way it does this is at the end of all of our linear DNA chromosomes, we have what are called telomeres. And so the telomeres are going to consist of non-coding repetitive sequences. So we purposely stick a whole bunch of non-coding sequences, just repetitive A's and T's and G's and C's at the end of all of our DNA. So that as we go through DNA replication over and over and over and over again, the hundreds of thousands of billions of times that we do that inside of our bodies as we are living our lives, we are going to protect the coding sequences from being lost as these cells divide. So as our body cells are dividing, we are losing nucleotides, so our DNA is getting shorter and shorter and shorter and shorter as we get older. We do have these telomeres at the end that are meant to protect us from actually eating into our actual genes in the kind of core part of our linear DNA. Now, some organisms have what's called telomerase. And we also have telomerase. This is an enzyme that extends the telomeres. Now, we see these in fungi and in protozoa. We also see them in human germ cells and stem cells, but not in adult somatic cells. Soma meaning body, so all of our body cells. So germ cells meaning those cells that are going through meiosis in order to make offspring, and then our stem cells, which are our original first starter cells to start all the different parts of our bodies, but we don't see them in our adult cells. So we don't have telomerase that goes in and it fixes those ends, which just as a side note, there's lots of research being done on the fact that they believe that that is a large part of aging. So our aging process is because we're losing these telomeres, and just the process of making new cells is eating our genes. And so our body does not function as well because we're starting to lose our actual genetic material to allow us to function. So there's a lot of research being done on, one, how to extend our telomeres, which they're taking a close look at that and seeing that that can possibly be reversed, but then also slowing down the cellular division in the body to try to elongate life or to maintain a healthier life for longer. So that's a side note. But with our telomerase, how that's going to work then is that if we have this DNA that's missing this chunk, because that's where the primer used to be, our telomerase is going to attach to this opposite this strand here, and then it's actually made up partly of RNA. And that telomerase is going to have the complementary RNA, the 3' overhang at the end of this chromosome. And what it's going to do is it's actually going to use this RNA template itself is an RNA template here of nucleotides as part of the telomerase. It's going to attach to the DNA here, and then it's going to use itself as a template to add more DNA onto our strand that had the primer. So we had the primer was attached to it, and then we have our growing strand was this one. Then what we have is the primer is removed. We have our telomerase that's going to come in. It's going to have complementary nucleotides. Then what it's going to do is it's going to add these additional DNA nucleotides here, which makes it longer than the DNA was. Then what happens is this telomerase is going to go away, and then what we can do is we can have a primer come in and attach up here, and then we can have the DNA being made in this direction. And then you'll notice that even if we go in and get rid of this primer, we're missing this DNA here, but that's okay because that wasn't even part of the original DNA. So we have the original DNA here, and then we just have kind of this extra section here, which is the extending of the telomeres. So the telomeres are very important, and there are various ways of eating and living and having different types of lifestyles that are going to help protect your telomeres from getting shorter and shorter. So we see this problem in eukaryotes. We don't see this problem in prokaryotes because, of course, their chromosomes are not linear, but they are circular. Good. So we are going to take another look at something that we see in prokaryotic organisms, and this is what we see in their replication of their plasmids. So remember, we have that extra chromosomal DNA in bacteria, and oftentimes the extra chromosomal DNA in bacteria, we have it in eukaryotic organisms as well, but in bacteria are plasmids. And plasmids go through what's called rolling circle replication. I didn't mean to imply we don't have extra chromosomal DNA. We do. Remember, that's in the mitochondria and the chloroplasts. But when we're talking about replication here of extra chromosomal DNA, we're talking about plasmids. And that is done by rolling circle replication. So this is used by plasmids. So plasmids. Also several bacteriophages. And some eukaryotic viruses is this rolling circle replication. So how does rolling circle replication work? So our plasmids are these small circular DNA, and I'm going to use plasmids because that's mostly where we see this. And what happens is we are going to start by nicking one strand at what's called the double-stranded origin, or the DSO. So we nick one strand at the double-stranded origin, or the DSO site. So we'll say that right here, this is our DSO site. So we go in and we cut it. Then DNA polymerase 3 is going to use the unnicked strand as a template. So use unnicked nicked strand as a template. So it's going to create one double-stranded DNA and one single-stranded DNA. So we have the DSO here, the double-stranded origin site. So our DNA polymerase 3 is going to go in, and what it's going to do is it starts to separate these two strands. So if we go in, we're going to cut right here at this DSO. And what we're doing is we're just nicking that outer strand. So then our DNA polymerase 3 enzyme is going to go in and it's going to attach to this unnicked strand. As it attaches to the unnicked strand, it's going to go in this direction around the circle, around the plasmid, and it's going to be adding those nucleotides, right? Let's not, however, forget that we need also RNA primase to synthesize a primer. Synthesize, that's an N there, synthesize a primer, so that we can have our DNA polymerase 3 start attaching these nucleotides to it. So if we go back to our circle here, we have our DNA polymerase 3 moving right along on this inner circle, and I'm not drawing it in the right place just because this is so small, but it's actually going to be creating the additional strand on the outside here, going along this circle, the unnicked strand. As it's doing this, however, this outer strand is going to be pulling away because this DNA polymerase 3 is going in between the two, and it's breaking those hydrogen bonds between the parent strands as it's making the other strand. So as it goes in and it's making this additional strand, it's kind of pushing off or displacing this nicked strand. So this nicked strand is going to be falling away as the DNA polymerase 3 is going to the other one. So we have what we end up with here is we have our parent strand. We'll say this strand is our other strand here that we went around and had, and then this strand has been displaced. So our displaced strand then has what's called the single-stranded origin site. And actually, of course, everything has that because that has to be replicated as well. So our single-stranded origin is our single-stranded origin site. And so we have our RNA primase, again, is going to synthesize a primer. And that's going to initiate replication at the single-stranded origin site. So it's going to make this single-stranded DNA into double-stranded DNA. So we already have our double-stranded DNA here because that's from our original nick, and it's going to go around and replicate based on that inner circle. Then we have our displaced circle here, and that's going to attach to our single-stranded origin site, and then it's going to go around and make this circle as well. So I didn't draw it very well here. Let me kind of back up a moment if I can so that I can make it a little bit more clear, because this displaced circle here is still going to be attached at some point. And as it's attached, it's actually going to be its own circle. So it's still a circle. Then we have our DNA polymerase come along and attach it, and then it's going to go around and it's going to make another strand, and then it's going to be attached here. So we actually end up with two circles at the end. So we have the displaced circle, and then that circle is going to have our synthesis of the second strand utilizing our DNA polymerase 3. So my drawing may not have been the best drawing here. I ran out of space as well, but you can see a drawing in the text that might be helpful where we have that original attachment of DNA polymerase 3 going around on the unnicked strand, displacing this outer strand here. And then that displaced strand is going to have its single-stranded origin site where the DNA polymerase is going to attach, and then it's going to make an additional strand on that single-stranded DNA so that we have two double-stranded DNA plasmids at the end, right? So remember that that's the goal. So we have one here and one here. They're both double-stranded. They both contain the DSO. So there's going to be a DSO on here and an SSO. So we have our two plasmids now, and then when we go through the separation of those cells, the binary fission part, then one's going to go into one cell, the other one will stay in the other cell. And then we have those plasmids in both of those cells at the end, the daughter cells. All right, so that's going to wrap up 11.2, which is our replication, our DNA replication. So we take a look at replication in bacteria and eukaryotes, as well as for plasmids or bacteriophages and some viruses here with the rolling circle replication.